So, good morning, everybody. I'm Anders Kästner, and I'm going to entertain you for this spring on the topic about quantitative big imaging. Nope, no, it doesn't work for some reason. Oh, no, it worked. Okay, good. So, today's lecture, I'm giving you an introduction about what the course is about the outline, how we're going to work, and so on. The whole course will be on this webpage where you have all the information. And actually, you can see GitHub in here. So all the course material is on GitHub, but I will come to that later. And all the lectures will be recorded. That's why I'm wearing the headset. So you will see, you have the possibility to actually check up on the lectures afterwards. And now these slides, they are actually made using a Jupyter notebook, and in some cases you will see some code here and there in the slides. Mostly it's just because I want to create a figure of some kind, but there are also examples that come up during the course where I want to show how to do it actually using Python. I come also more to the topic Python, but for now, each notebook that I'm working in needs some libraries to execute, so I need to actually execute this line. And now we are ready for the lecture. So about the course, a little bit presentation, who we are, what I think you are, and what is expected in the course, and also why the class exists at all. And then we go into more about images and a little bit about how the computing work with computers have changed over the years. And yeah, that's about it. So first of all, as I said, my name is Anders Kästner. I work usually not here at ETH, but at Paul Scherrer Institute as a beamline scientist, producing a moderate amount of images. And here I'm now been giving this class since, I think, 2020. So I have done it a couple of years. And before that, I was algorithm developer at Varian Medical Systems. So I did a couple of years as a software engineer. And before that, I was here at ETH as postdoc at the Institute for Terrestrial Ecology. I think they renamed now to Environmental Sciences. It's just a couple of buildings in that direction. And then before I came to Switzerland, I'm actually Swedish, so I did my PhD at Chalmers Institute of Technology on the topic signal processing towards images. And there you also have my email address at PSI. I also have an ETH email, but I prefer if you contact me on that address. I check the other one also occasionally, but not as frequent as this one. Then we have Daniel sitting here in front. He will support you during the exercises. So every week we have 1 hour of exercises after the lectures where you can try to work with images using the topics we have talked about in the lectures. And you will hear and see more of him soon. So, who are you? I looked in the list of enrolled students, and to some extent, this cloud actually covers what you are. Some mechanical engineers, some from electrical engineering, informatics. Actually, I have a mathematician also around us, physicists, and so on. And all of you have different background, different levels of programming skills. So I'm not relying on any high programming skills. That's not the class for that, but there will be a line of code here and there, and also the exercises. It's more about putting up recipes piecewise. Also experience in type of data you work with, the workflow, and so you can have, some of you may have rather simple data, other ones data with high complexity. Some prefer working with user interfaces where you can click and mark up things. Other ones prefer working on the scripting way. Other ones see these things for the first time. Other ones already have some experience. Some may even have quite a lot of experience and come to a dead end somewhere, maybe. So the idea is that I'm going to show you how, in principle, how things work. I'm not going deep into the math of image processing. Then we'll probably need 2 or 3 times more time than we have now. Also, for the exercises, we are preparing notebooks where there are some code snippets, and then you have to adjust them, maybe add a few lines, but it's not about writing advanced classes for image processing or something like that. That's not the idea. It's more about getting grasp and see what happens when you apply different operations. And well, the exercises, there is usually 1 per lecture. They are more focused on the first part. The second part, we spend more time on a project, which I will mention on the next slide. And we are using mainly Jupyter notebooks for the exercises. Has any one of you already used Jupyter notebooks to some degree? Looks like a lot of nods. That's good. So you know the basic concept about them. If you want to go on the advanced level and doing fancy stuff, you can write your own Python classes or Javas or C++, whatever you like to do. Actually, I don't recommend doing the exercises in C++. It's a bit messy, to be honest. It's possible, but I don't recommend it. Java and Python. Python is more or less the language we are using, so it's more taking the code out of the notebook and put it in a file on the side to get a cleaner workflow. Then there is the possibility of doing a project. So if you have a task in your master project or in your PhD project that you want to work on, this is an opportunity to try the methods that you have learned here in the class with our supervision, so you can get hints, ideas how to progress. Also, if you don't have a project, we have some examples that you can work on, and also try the methods anyway in that way. And I say it's not mandatory, it's actually optional, but to get the most out of the course, it's good to do them. Another thing is, I'm actually also giving the opportunity, because this is extra work for you, so I'm giving you the opportunity to use this project as a starting point for the exam. The exams are oral, so it's a discussion where we discuss through, I ask some questions or show an image, and you tell me how you would work with it, for example. And if you then do a project, I'm using questions. I'm making questions around the project, so you have a running start for the exam. But the important thing is that you try and get your hands dirty on some data using the methods you have learned. Of course, last lecture opportunity is actually not me talking, but it's a presentation day. So those of you who did the project, you can present how you work with it and show some results from your analysis. So this is the overview of what we're going to do. So today we have the introduction and workflows, and then we're going through different topics. So next time we talk about data sets, then we're talking about how to improve the images, how to start extracting some information from the images, so basic segmentation, which actually continues with more segmentation. Then we start analyzing what we have segmented, and that even continues a bit. Then we switch over to statistics, where we are looking at uncertainties in the analysis and also statistical analysis. And then we continue with some topics around dynamic experiments, which is a special type, which also requires some extra information or workflows. And then in some cases, it's not enough to use just one type of imaging, but you use several, and then multimodal imaging is about how to combine these. The last lecture is scaling up and going towards big data approaches, how to work with larger data sets. And well, then there is the wrap up and project presentation to the end. So this is how it's going to work. And there is some reading material. This is, say, complementary material to what I'm saying. And it's well-readable information here. And also, there are some useful literature. For example, this John Ross Image Processing Handbook. It's a very thick book, I don't know, over 1,000 pages, but it gives an overview of the topic image processing. Doesn't go down in the details. For that, you need to go in the references he pointed at. But it's a good starting point. And then we have some other relevant books. So, for example, hands-on image processing with Python. Yes?
So I don't know anything about machine learning and image processing, but I think there's like traditional image processing and machine learning.
Yes.
So when would you use machine learning and when would you use image processing in the classical way? And so what will
Depends on the task.
Okay, and what will we learn in this course? Is it more traditional? Or
We are mainly going towards the traditional one, because to be able to select the machine learning choice, you can do it blindly, but then it's really a black box. And also the machine learning based. So for example, if you go for a unit image processing, it actually contains a lot of the stuff that I'm talking about. So you have a better understanding what the machine learning is doing, and possibly even why a little bit. You can even look into the weights and see what happens in the different weights, and you can see what it grabs on and weights together to get the result. So I'm going to show a small example using a unit during the lectures, but machine learning as such, well, machine learning is a new label on old stuff, in principle, with a lot of new additions, but simplified, it's a lot of statistical methods, all the regression types and everything is everything bunched under the name of machine learning. So we touch a lot of things that approach the way you work with machine learning. Also the workflow, how you work with validation and tests, it also applies to the traditional image processing, because you want to know how well you have worked. If it's you who did the algorithm or the machine did the algorithm, you still want to know how well it did. So it's kind of nested together.
Also, it's basically a good foundation to be able to do image processing regardless of the tool you will use in the future.
So I'm more into the workflow, seeing what obstacles you come and ways to solve it. So I also don't have the time to go deep into machine learning for image processing. That's a class of itself somewhere at ETH, I believe. So there is no time for it. It's call it a pragmatic approach. You have to get the scientific method to go through practical experimental data. Thank you for the question. It's good. So, the motivation, actually. Now I come to that part. You have data. So, the procedure is usually, when you want to do an experiment, you need to get access to some kind of imaging equipment. If you have it in your lab, then it's quite easy. But often you have to apply for the resources, maybe at large-scale research facilities like Paul Scherrer Institute, where we have several different imaging opportunities. Then you even have to write a proposal to get your beam time, and then yay, you are allowed to have 2 or 3 days of experiment. And that produces a lot of data. And you have to do something with this data. And then you come to the confusion part, what to do with it. If there are some gigabytes, it's quite easy, but some experiments even result in terabytes per day. And then is the question how to handle it, because you don't want to sit there and click on every image. Then you would be gray-haired before you're done with it. Also, we want to understand what's in the data. And in the beginning, you more or less only have data. So you do some acquisition, you do a reduction, transforming it in some way. May also do some reconstruction. And then you apply some filters, but you're still talking, it's only data. There is no interpretation of what you see, except your own visual qualitative information that you see, okay, it's getting better, I can understand something in it, but you haven't quantified anything. Next step is you go for the segmentation. Then you have already divided the image into some pre-knowledge, and then you start labeling. You have identified different regions as, this is that one and that is that one. And from the labeling, you can start doing measurements. How big is it? Is it curvy? Is it round? Is it, more like blocky, whatever. And you start measuring. And with that, you start introducing more knowledge and understanding what you have in the image. Then you can do some classification, saying if they are round, they are that class. If they are squares, they are in that class. And then finally, you can also do some statistical inference where you really have, let's say you have many data, different test groups, and then you can start making conclusions about what's happening. And then we are talking more about knowledge and understanding of what this data actually includes. And the question is how to proceed. And many beginners, they just click around, test a lot of things. They find, oh, that method is funny. I tried that, and then you do another one, and then you do another one, and then you realize, oh, yeah, I need some math. Oh, yikes. And then I have to go back again. And we don't want to have a workflow that looks like this, because you should know exactly what you do, step by step, and why you do it. And it's also important that this workflow should hold not only for one single image, the test image that you work with days and in and days out, but it should actually be so generalized that you can do the same thing for 10 images maybe, for 100, even 1,000 images. And your work effort shouldn't be increased by the same factor. The computer will work that factor, for sure, but you shouldn't. So ideally, you should just need a couple of few parameters that you have tuned, and then you can just let the computer do his thing for I don't know how long. And then afterwards, you tap off ideally, some nice graphs, but maybe you need to do that in some intermediate steps. But anyway, the main big processing should be as automatized as possible, because otherwise you will go crazy. So the experiment life cycle is like in most scientific experiments. You design your experiment. First of all, you need to have some hypothesis. From that one, you design an experiment that can validate this hypothesis. And that's sometimes can be very time consuming. Then you do the measurements, and as I said, if you get beam time at large scale facilities, that's very intense time, probably 24 hours a day over a few days. So make sure you have enough people to handle it. Then the next step is the data management, and how you actually the management should even be planned already the experimental design, but managing how you will handle your data, how you organize it in folders, if you have tables of some kind that identifies it. So you need to put up some databases that can make automatization easier. Even a simple thing like the file names of your images can be a deciding point if your data is easily or not easily analyzed. I have seen it a couple of times, people that put the metadata as information in the file name, and that's horrible for mass production. So please don't do that thing. I've seen like projection angles as part of the file name. Yeah, that's not a good way to store the data. Metadata should be either in a table on the side or written into the files. And then you come to the post-processing, which is also an extremely time-consuming task, in particular if you have a lot of data. But with the help of data science approaches, this has evolved quite a lot over recent years. So there are a lot of tools that can help you to handle your data in a better way. And if you look at how much time is spent for the different parts of the experiment, you can see that around 2000, then the experiment design was quite, how many percent? Like less than 10%. Then you did the actual measurement, which took a remarkable part of the time. Data management didn't take that much time because back at that time, you didn't produce that much data. There were no streaming detectors, and experiments were a lot slower. And then you did some post-processing. And with time, you can see that the experimental part is shrinking to almost nothing, because today's experiments, as I said, can produce terabytes of data in very short time. Then you have really a problem with managing how to take care of the data, how to store it, how to copy it. Just copying a terabyte of data takes time. And then, of course, you have the post-processing, which also takes quite some time. Experiment planning can maybe, depending on how you set up the experiment, can be more or less. But it depends also about the complexity of your experiment. And I already mentioned we have today detectors that produce a lot of data. I think this number has been exceeded now. So, but anyway, we can talk about, as a guide number, 8 gigabytes per second. And already that is quite an obstacle to handle. You need high performance communication lines to bring it down into fast streaming storage units, and then afterwards, the question is what you do after some seconds. And just looking at that, you have, if you work with a workstation, you have saturated the memory in short time if you are working with data of that kind. So you need ways to handle the data in that sense. So one thing is having larger clusters if you want to process that amount of data. And that needs also to be planned in the whole workflow that you have. Otherwise, you will be really overwhelmed. And then there's an interesting snapshot. Some camera that produced in 2013 3 times more images per second than was uploaded on Instagram. I don't know what it is, the status is today, but I would expect it has changed a bit. But anyway, at that time, there was a camera that was pretty productive. Then we have different sources of images, depending on what you're doing. So you have X-ray, which is used in medicine a lot, but also in material science, biology, and so on. So it's not only the medical application, even though that's probably the first what people think about when you say X-ray. And you have even special acquisition techniques like CSAX diffraction that can produce 30 gigabytes of data per second. And then we have a nanoscopium beamline, which produced 10 terabytes per day, and also huge data files that it works with. You can go on to microscopy, and then the image rates or data rates are not that high. But you can even go down to the personal equipment. So you have, for example, the GoPro camera. This is also old data, but it would, in principle, be able to produce something like 60 megabytes per second. And it's not very expensive. I also yesterday looked at cameras that are developed for astronomy. They also produce one image is 50 megabytes, and it can do it at a frame rate of 14 frames per second, which is also already quite a lot. And that camera costs only 2,000 francs in that order of magnitude. And it's used, it's getting more and more popular, at least at neutron imaging beamlines. And yeah, so there are many data sources that produce images. Yes?
So for the GoPro, like 3,840 times 2,000 times 30 frames per second, can you instantly tell the megabytes per second from that, or how do you?
It's not a fair number because it compresses automatically. It in hardware, it already compresses into some MPEG compression. So but if you would look at the raw data, you would expand the film, then you would get something like this.
But there's like the formula which is like straightforward to put in the numbers and you know how much megabyte it is for that.
I would guess so, but you can also set the streaming quality, which resolution you want to use. So you can tune it, but I believe you can compute it somehow. They probably have some table, but the size of the output file doesn't really reflect the raw data size, because it actually does a lot of hardware compression, and that's done in most consumer electronics, that they do compression. And talking about compression, there are different kinds of compression. You have lossless and lossy compression. And the MPEG format is a lossy compression, so you lose some information in order to compress the data even harder. Otherwise, the lossless, I think you can gain about 30%, not much more. So that would be the same as zipping. But it's in that order of magnitude. But using lossy compression, depending on what you can accept in image quality or film quality, you can do quite a lot. So another thing is how to handle the masses of images. And when you do the processing, you actually increase the amount, temporarily increase the amount of data you have. So first you do your acquisition, and then you do a reduction step and a reconstruction step. Then you don't reduce the number of pixels. So you're actually adding up, because sometimes you want to save what you did the previous step to be able to go back only one step instead of doing the whole process. So it linearly increases up to a certain point, and that is when you go from filtering to segmentation. Segmentation means that you go from maybe 16-bit or floating point data, which takes 2 or 4 bytes, to maybe just black and white. And with that, you have already a starting point in data reduction. And then when you do the labeling, you can go again, also go into less data. And when you start doing the measurement, then it's when you go into the real data reduction, because then you have identified items, and then you will only have a table saying item 1 has area that, volume that, and. And that's a radical reduction in data. So ideally, you should go from that point to that point as fast as you can, because it's much easier to work with. That one requires large workstations or even clusters to do, while this one can be done on any laptop. So you need to, so the idea is you should process, go as fast as possible to the small data amounts. You may have to go back again, but then you have the large data again. So I have been talking about terabytes, and if you looked at an image like this is just noise. It's 1,000 by 1,000 pixels, 1 megapixels every second. Then it would take you quite some time to look at it. So if we look at this example, we have 1 terabyte, which is 1 to the 10 to the power of 12. The image size is something like this. And then an hour is the number of seconds. So if you do the calculation here, you can see that to watch an image per second, it would take you 140 hours about to view all the images of a terabyte. That tells immediately that it's not really possible to do the manual inspection. You can look at samples. You take one image here, one image there to check the quality, or actually even to test. But really looking at all images, there is no chance, and even less to do any analysis. Well, this would be, in principle, just swiping from image to image, saying good, bad, good, bad, good, bad, or something like that. But 140 hours, I know better things to do in 140 hours. So now, let's say we want to look at these images. And this is some bone cells in a slice from bone. And we're looking at some cells in this slice. And right now, you only see white blobs in this black background. And with some different questions you could ask here. So first of all, ignore the ones that are too big. That would be maybe this one and those. They don't have the right shape. They don't actually match the pattern. Probably it's just artifacts from the segmentation. And then you want to do some counting of how many cells you have. And then maybe you want to do some, say, telling if there are more on the left side and then on the right side. And even tell if they're bigger on one side than on the other side, or top or bottom. And all these questions should be answered from this image. If you do it manually, you will probably sit some minutes to make the conclusions, looking at it, and doing it with image processing would just be like that. It would tell you within very short time. So for sure, the computer would come really into use here. But that was only one single slice of the bone image. Normally, when you look at this kind of data, you have volume data, and of course you want many, because you want to look at the statistical variation between different individuals. And in 3D, you get maybe 2,000 slices, so the data has grown really a lot. And then doing it really bad is you want to have different species or different individuals, and then you come up in a sample size of about 1,000, which is kind of typical for biological investigations, that you need that amount of data in order to make relevant conclusions. And now is the question how to do the measurement here and how to analyze it. As you see, this is actual data, and that was actually my predecessor in this class. He had it as his PhD. He even designed a robot to move in samples in the scan and doing all these scans, and then afterwards, afterwards he had to analyze it, and that was more or less the starting point for giving this class. And then we come to even more metrics that makes it also difficult. Some metrics, they are really easily measurable, putting a number on, but there are more fuzzy measurements too, so a level of alignment or some kind of grouping between different clusters, and then it gets even more difficult because you need to define some kind of metric that can tell you this information. So here we have fuzzy classifications, kind of aligned, well aligned, or not really aligned. And getting into these fuzzy classes and let the computer tell you them is not always so easy. By eye, you can easily tell this one is good, bad, or different, etc. But often the frustration comes when you have to tell the computer to tell the same. It's so obvious to you. You see it on the screen, but it's not always that way. And another part that makes it difficult or produce even more data is when you have dynamic information. So you make a time series. In this case, we have a little video of some foam where you see the bubbles move. These are very coarse steps. But questions that are asked here is first, how many bubbles are there? How large are they? Is there size distribution difference depending on where in the sample they are? Lower or in the top, along the walls. Where do they move faster because of the fluid dynamics? And also if they are rearranging, maybe merging or splitting. All that information we want to tell from a data set like this. So, until now we have only talked about images. I mentioned that we want to use them to validate an idea. So that brings us to the scientific method, where we have, first of all, we have to define a hypothesis based on maybe some previous observations. Then we produce a hypothesis, and then we want to produce an experiment that we can convince other people that this hypothesis actually is valid. And the first step what you grab for is maybe the qualitative assessment that, well, I can see the observation here, which seems to be right, but you can't really prove it, and in order to do that, you need to extract quantitative information. And in our case, our tool to measure is the images, and then we need to have reproducible way to analyze these images to make sure that it actually happens, actually can be convincing people. And this analysis is far less complicated than in other cases. For example, if you measure the temperature, you have your thermometer, and of course it reads out maybe 50 Kelvin, and you can look on the back and see, okay, with that accuracy or that uncertainty, and then you have it. And then you can do a couple of measurements of that. But in images, it's very easy to do, you have to do a chain of operations in order to get your measurements, and depending on how you choose them, they are more or less robust, and that makes it, can make it very confusing how the analysis comes out. And of course, there are also a lot of methods to choose from. Every image processing lab have a bunch of their own algorithms that they develop for different purposes, and there are also lots of tools. Probably every student starts writing some of their own little toolbox, and some parts of them may be useful for my project as well, but it's about finding them. And of course, we need a multi-step process in order to handle it, and testing and finding the right method, not only the experiment itself, maybe you need to iterate the experiment, and then afterwards you have to iterate the analysis procedure as well, and that is all time consuming. The experiment itself probably cannot be repeated too many times, but the processing, maybe your computer runs hot for, say, an hour before you get your answer, and then you see, oh no, that parameter was wrong, and then you have to reiterate, or that equation was a bit wrong, then you have to reiterate, and then it takes time to get the right result. And important questions, very often, it is nice to have a 3D rendering. And these examples are from studies in cultural heritage. That piece is some gold piece that was found in Sweden from the Viking Age, or pre-Viking Age, I think even. And here they wanted to do more or less a visual analysis because they only had one sample, and they want to see how it was made. For that purpose, you can go for the visual analysis, but often we come into more complicated cases. So here we are looking at corrosion and treatments, how to reduce corrosion of iron, which is a very relevant topic for the archaeologists who want to stop the corrosion as much as possible. Otherwise, all the nice items they find, they just corrode in a few years when they excavate it. And here there was a study where we looked at how the corrosion, or how the intensities varied with the corrosion or the product type in the material. So, in order to make or plan the experiment, you first of all have to make sure, what am I looking for at all? And then you may have to reiterate that. Maybe the original question is too general, or maybe too specific. Is it possible to really conclude anything regarding your hypothesis on it? Then a very important question is how I can plan my experiment in order to make the analysis easy. Because everything is a unity, and if one part makes the other one harder, then you may reconsider how you did the experiment. And also, very important question is, how can I transfer my original question, which is related to my special topic, whatever that is. Could be something in medicine. I want to see if an organ is sound or not, or if, as in the previous example, if the corrosion is stopped when I do this or that treatment. But those are the questions that are domain-oriented. Now we make images, and we want to know what operations can I use in order to make this analysis. And then it's always good to have a priori information, because any analysis method, if it is the traditional way or if it's machine learning based, the more a priori information you have about the sample, the better. And in particular, if you can use it. Some information is irrelevant, then of course don't bring it in because it's just confusing. And the purpose of the experiment could be compare different treatments, could also be to extract modeling parameters. So for example, if you look at the water uptake in soil, you want to have some transport parameters that you want to quantify on that particular soil. Could also be that you want to inspect and characterize some sample. Was this process successful, or did it introduce some faulty regions? And in that case, where are the hotspots in these faulty regions? And sometimes it's even possible to do a 3D visualization, but that is more eye candy, mostly for the presentations, because it's always nice to have eye candy in the presentations. But in some cases, 3D visualization could also be a way for the qualitative analysis. And what can we measure? Depends, first of all, on what information you want to measure on. You may want to look at the gray levels, changes in gray levels, which can be related to material composition or material transport. You can also look at shape quantification, where you look, first step is just to identify items and count them. But once you have the items, you can look into measuring volume or shape characteristics, etc. So, why quantitative? One thing is the human eye has issues to see what is good or bad, what things are. So in this example, it's a classic visual illusion. If you look at these, which of the inner boxes is the brighter ones? Which one? No, it actually has the same intensity. That's a visual illusion, because depending on the context, the eye thinks, it's brighter. But in reality, it's actually the same. And seeing that it is the same is something you can only do when you measure it. Another thing is when we look at this gradient, or stepwise gradient, it looks, there's an, say, perceptive illusion that around these steps, that the intensity goes up a little bit. Do you see it? And that's also the same effect, and that's also pretty annoying when you start doing the visual analysis. It looks really like it goes down and up, but it's really sharp edges, sharp steps like that. And if you have gradients, then it can even be more tricky. So you need to select the right tools to do your analysis tasks, and you have a catalog of criteria. So first of all, how many samples do I have to analyze? How complex is a sample? And is human interaction or interpretation needed? And in that case, at what point? And in some cases, people say it is needed that every selection is done by humans. In other cases, like in machine learning, you have to create training data because the machine learning is more or less based on having a ground truth and or some input or the expected input. And in this combination, you need to do some pre-labeling, which is done by some experts. And when they are done, you just buy the data and work with it. Also, what is the end product of this? And maybe there are even tools available for this purpose. So in that case, use them. And the last question that I put up here is, will there be similar data sets in the future? So maybe it's an experiment series that you're doing, or your lab are working on more or less the same type of images. And that goes into more efficient workflows for the whole group, for example. Given this, you have the choices working with interactive tools. So as soon as you need a human interaction, then of course you have to go for some interactive tool. Then you can do scripting using existing toolboxes, and that's quite helpful. If someone already coded it, it's hopefully already validated that the code is doing what it's expected to do. And in last case, you may have to develop new algorithms. And I would say, think twice or even 3 times before you're starting this, because that is a hole where you can spend a lot of time. So maybe you check once more, did someone else do it, actually? Can I use something at least before you start developing new advanced algorithms? And for this, we want to look into actually reproducibility and repeatability, and what the scientific community in principle really strives for is to have reproducibility, that someone can take your paper and reproduce exactly what you're doing and come to the same conclusions, plus minus some uncertainty. But what we more are looking for is repeatability, that you can take the same kind of data, let 2 persons start the process and do the analysis and get the same result. That is at least what you should strive for. If you can achieve that, it's fantastic, but this is at least achievable. And yeah, that's more or less what I said already. Well, it's not so easy to do. You actually need these the repeatability to be able to handle the large data sets. And with image processing, if you need to change a parameter for every step, then it's harder to get the repeatability. So try to get automated algorithms that work well. And very important is to make, for the repeatability, is to keep track of what you're doing. And the first thing is document every step you do. If it's code, document how to use the code and which steps you are supposed to do in order to achieve a certain result. And something that is in the beginning very hard to keep is to write clean and understandable code. In the beginning, when you start, sorry using the expression, but starting dabbling around, writing some code for the prototype, it's usually you're happy when it runs, but you change a little bit here, change a little bit there. It's not always so easy to get an understandable workflow from that. So once you have a working thing, the best thing would be actually to rewrite it and clean it up in order to make it transferable to someone else. And there are different ways how you can approach the image processing task. And in the beginning, what you have is more or less just a bucket of pixels. It's just a matrix of numbers. That's all it is when you start. And then depending on what background you have, you do a different approach on how to analyze the data. So we have, for example, the experimentalist approach, computer vision approach, and deep learning approach. And they approach how to work with data very differently. The experimentalist goes problem-driven, top-down approach, and usually there is a reality that they try to model and follow. And examples would be looking into cell counting or porosity measurements or something like that. That's really related to what the experiment is about. If you go for the computer vision approach, then you have a more technical, method-driven approach. So you look for features in the image, what can you filter, what can you segment, and you look into more a signal approach on the solution. And that could be, for example, you want to look at edge detection, face detection, or something like that. And finally, we have the data scientific approach, which is result-driven. So that mostly requires that you have a ground truth, and when you feed the data through, then you see how then you more or less have told the system, learn what I need to have in to get this out. And that could be, for example, image captioning. So it sees an image, this is a cat, this is a dog, or something. Or also to identify unusual events could also be an idea. And biology inspired is meant just that the learning procedure of these machine learning methods is a little bit what's happening actually up in the brain. So we are talking about neurons and how they are connected. And with that, I think we make a break. A little bit delayed, but I think this is a good point to change and make a break.